Dr. Dillon, I think you're muted.
Sorry, can you hear me now?
Yeah.
Sorry, I think my headset shut off on me. All right, well, let's start over again. So we're going to finish up our last lecture, which seems like a long time ago now at this point with the intervening holiday. So hopefully you guys had at least somewhat relaxing spring break and maybe caught up on some sleep or did something fun. So we're going to finish up talking about the human example of animal microbiomes. I don't focus a lot on this for this class, but we will touch on it. And then we'll start talking about plants. So as you guys are hopefully aware from the announcement that I made, right? So you're either going to come on Wednesday or next Monday, not both. So you'll turn in your outlines by 11:59 tonight, and you'll send a copy to the partner that I posted. So if you don't know them and you don't have their email, just send it via the Beachboard email function. That should work for you. Or if you want a direct message in the chat to each other and share your email, that works too. So the idea is you guys will give each other feedback, and then you're going to meet with me, just the three of us. Each pair I'll meet one-on-one, and so that's why there's scheduled times. We have more students coming this week than next week, so we'll have about 9 minutes on Wednesday. So I know it may not seem like too much time, but I try to give a pretty good set of feedback during that times. The more that's in your outlines, the better off you are in terms of getting feedback from me and your partner. Okay. So as you finalize your outlines to turn them in tonight, please, think about that in terms of if the examples I give in the rubric are very simple, right? You can turn that in and that's kind of the bare minimum. But if you give more like sentences or, you don't have to write paragraphs, okay? You might have a paragraph for the intro, but you're really not going to use paragraph form for this outline. But the more information that you put into the outline, the more feedback you can get from me and your partner. And that's going to only help you to make it a better final term paper. Does that make sense? Any questions about the expectations for the outline due tonight? Okay. Hearing none, we'll go ahead and get started then.
I have a question about.
Okay, go ahead, Johan.
So for the references, I'm having a hard time making it perfect. Is it fine for the outline? Because I use a manager, but it's still not perfect. Like you use AMA, right?
So it's the one that I had on there as the example, I think, was Developmental Dynamics Journal. So every journal has a different format. And so it's in those reference managers, you should be able to select. But you can have it formatted using that reference software, and then you can always usually you can go and tweak it afterwards. I'll give feedback on it from the outline, and if it's not perfect, it's not as big a deal for this as it is for the final term paper. What I'm most concerned with is that you have at least 10 references in this, and that you get started on, learning the referencing. So, yeah.
Thank you.
Okay. Let's see, something else in the chat. So Angel asks if they all need to be primary literature, if they can be secondary or tertiary articles. So the idea is it's mostly primary literature, most of them from the last 10 years, but you can have some earlier citations, especially if there's like a seminal paper written on your topic. You can use review articles, right? And in fact, review articles are a good place, as I said in the rubric, for you to start with, right? But I don't want you only citing review articles or some of these more derived things, because then you're not getting to the original studies, right? So we want to primarily, so I would say if you're balancing it, you want at least 2/3 to be primary literature articles, okay? The way you cite those may be very different though. We're trying to be integrated here. So I want you to, you know, you're not like summarizing one paper in a paragraph and then the next paper and then the next paper and the next paper. The idea is you're telling a story, you're citing multiple papers at the same time on various topics that you're going to be outlining for me for tonight. Okay. Some citations, you're going to only cite it once for the entire paper. Others may be cited repeatedly because they're really important to the arguments you're making or the topics that you're describing. So not all citations are created equally, but if it's going to be in your citation list, it needs to be something that makes a meaningful contribution at least once in your text. Does that make sense? So you're typically going to have multiple citations for a given paragraph. Okay. And so you're going to cite those as you go along. Okay. In the final paper. For now, you're going to put them like if you have a sentence of this is the topic I want to talk about, like subtopic 1-3, then that will have a citation at the end. So I know you've got a paper in mind or 2 papers or 3 papers that address that issue. Okay. And again, I'll go over that, but hopefully that answers your questions. We can get into the specifics of your individual outlines on Wednesday and next Monday. Okay. All right, so I'm going to share a couple of examples. We'll talk about gut and the oral microbiota. Right, so in terms of the ecology, a big part of the ecology of the gut has to do with pH, right? And sort of the habitability of these various parts of your intestinal tract. And so as you can see here, the diversity tends to go up as you go down. Because of all of the essentially winding intestines, there's a huge amount of volume and a huge amount of surface area. In fact, there's larger than several tennis courts, as it says here, right? And so as you move from the high acid of the stomach and you move down towards the colon, you get more cells and you get a more diverse assemblage. So it may just be Helicobacter up in the stomach because of all the acid and they live in the lining of the stomach. But then the small intestine will have a higher pH. So it's got, you know, more dense cells and more diverse cells. And really the colon is where you end up with a huge diversity. All right. And this is anaerobic as we talked about last time. Okay. So just like with the cow rumen, human guts are very diverse and they play a huge role in human health. Okay. So what are those nutritional roles? So metabolism is a huge thing, right? And so anaerobic metabolism, because we said that the gut is anaerobic, is going to be the main thing. So what kinds of things are going to be produced there? What do you remember from the rumen? It's going to be very similar. What are the major products of some of those anaerobic metabolisms?
Methane.
Okay, so methane is produced, and at one point I think I had an estimate that about half of all people have methanogens in their guts and the other half don't. So we're not cows. We don't have to have the same assemblage as them. What about bacterial metabolism? What kind of microbes do you think we have? What are the major processes that are going to go on in the anaerobic gut?
Maybe some fermentation?
You can bet on that. Yes, fermentation is going to be the dominant metabolism. And so what were the products of fermentation that we talked about with regard to the rumen that might come into play here? Does anybody remember? We called them VFAs, right? What did that stand for?
I think it
Volatile
was
fatty
volatile fatty acids.
Yeah. So those volatile fatty acids, and as you remember in the rumen, it was really important for them to transition over into the bloodstream so that it raised the pH of the rumen. We like the rumen, it's a little bit different, but basically those volatile fatty acids can transition across the intestinal wall as well. Okay. So they become important. We get carbon from them. We also get vitamins, and that'll come up in a second. So microbes are going to make up about 30% of fecal matter, right? Fortunately, unlike those lagomorphs like rodents, we don't re-ingest our feces, but you can see why they do, because the microbes are there and they can, be eaten. We don't do that. But the microbes, of course, play a huge role in helping us digest food, in addition to the acids in our stomach and the peristaltic action and muscular contractions and things like that. They help to bring things down. As I already mentioned, we do absorb the VFAs that are produced. And some key vitamins like vitamin B12 and vitamin K that we can't produce ourselves are produced by coliforms and other gut microbes. There's also bile acids that are important in steroid metabolism that get made by your gut microbiome. But we're only now discovering, with the revolution over the last decade of the human microbiome, and gut is a big part of what those studies have looked at, we're only now appreciating the effects on nutrition and the roles that those microbes play in things like obesity and other sort of nutritional issues, right? And of course, they produce gases like CO2 and methane in some cases. Hydrogen sulfide, there can be sulfitogens, right? Sulfate reducers that live in our guts too. And of course, we now have an understanding that our gut microbiome has a network of interactions across our entire body. So they're now finding associations between gut microbiomes and neurological disorders. So really, we're now appreciating that the gut body interface and our microbes play such a role in that is really complex, and we don't understand a tenth of it at this point. But we now have the tools to begin to unpack this. The other major arena I want to talk about is the oral cavity, right? So as we all know, when we go to the dentist and they clean our teeth, right? After you come home, you can suddenly feel your teeth, right? And all your individual teeth. Before that, you had just kind of like this smooth thing. And if you stick your tongue in the back of your front teeth, right now, unless you went to the dentist this morning, you can feel that slimy biofilm that's formed. So oral cavities have a hugely diverse population, just like the gut. And like the gut, it's fortunate we can culture a lot of them, right? Not all of them, but in the mouth, we've cultured even more than the human gut, and we've cultured about 50% of the different groups. But some of them are as yet uncultured for sure. Again, these are anaerobes, right? Which might seem a little weird, right? It's not like the gut, right? In fact, we breathe oxygen in our mouth. So why are there anaerobes in the mouth? Why do we have so many anaerobes in the oral cavity? Where do you think we find most of the microbes in your mouth? Are they floating around in your saliva? Or where are they living? So Jenny puts on the teeth or the tongue, and Jocelyn says in biofilms, and those are all correct. So your biofilms all around, but they're on surfaces, right? Otherwise, they just get washed down your throat into your gut. So the idea is that these organisms have adapted to live on the surfaces. And of course, our teeth are like major surfaces, and your tongue is a major surface. It turns out that inside biofilms, it goes anaerobic very quickly as you move down, even like microns down into a biofilm, it can be anaerobic. The other reason you want to be in a biofilm is saliva is actually not a great food source, so to speak, and it can actually have antibacterial properties. So there can be lysozyme and lactoperoxidases and other oxidative enzymes that are in your saliva, which will kill bacteria. But if they live in a biofilm, then they're more resistant to those defenses. And so microbes are primarily going to be eating on the foods and beverages that we eat and consume, right? If you had a Coke before you came to class today, right, or another sugary drink, then all that sugar that ended up in your mouth can be metabolized by those microbes. And of course, that leads to things like cavities, right? Your epithelial cells, your cheek cells, right, which I'm sure we've all swabbed them at some point in some class or in the doctor's office, right? They're also a food source nutritionally, and they're constantly being sloughed off. And so when we talk about the biofilms on your teeth, you're primarily going to be talking about plaque, right? That's what a dentist calls it when they scratch that off. But basically, that's a biofilm, okay? And so biofilm formation, after you go to the dentist and they're all like clean and white and you've got no biofilm or very little biofilm left, very quickly, within a matter of hours, you're going to reform some biofilm. And initially, you've got your saliva laying down a glycoprotein layer, okay? And that's shown kind of here at the tooth surface on the enamel. And then you're going to get colonization by things like strep, right? So we all, you think of strep, strep pyogenes, strep throat, when you're getting sick. But remember, there's many species of strep, and many of them are normal occupants of your mouth and your throat, and they're not causing strep throat symptoms. So these strep are often some of the first colonizers, but then they pave the way. And here's that word we talked about before, remember, succession, the change in community over time. And so this creates an environment where others like fusobacteria and spirochetes and actinomycetes and these other diverse groups shown here by all of these there's spiral shapes and fusiform shapes, etc. And these all live together in the biofilms, right? So who remembers what we called this kind of succession, where the arrival of some organisms makes it more likely that others will come later and kind of supplant them in the... Anybody remember?
Is it facilitative or facultative?
Yeah, so it was facilitative or autogenic. Remember, so like your actions lead to that. In some cases, it may lead to you losing that. They may eventually be replaced. And I see, Hamza, you put in there, is it strep mutans? So strep mutans is one of a number of strep cultures that have been isolated from mouths. But so the strep actually stick around. They don't get completely excluded, but obviously they come into competition with this diverse group. All right, of mainly anaerobic organisms. All right, and then of course, it's a love-hate relationship, because this is a natural thing, but of course, if you end up with cavities or periodontal disease or things, oftentimes you get bacterial infections that then need to be removed. And even to this day, the dentist, or you may be sent to a specialist, will often have to use bleach to try to remove your biofilms. Biofilms are highly resistant to antibiotics, so you can't just take an oral antibiotic and get rid of your oral biofilms. So they actually will irrigate with, pretty strong bleach solutions to try to clear out. Like if you have a root canal that gets infected and you have like an endodontist that needs to go in and do that. All right, so that's it for that. Any questions about human microbiome stuff? All right, if not, we will move on. And Johan, I see you put in there an interesting link. Yeah, so there's a lot of stuff that's emerging in terms of this mind-gut axis. It's really, really fascinating work. All right. So we've talked about microbes in animals, and now we're going to talk about microbes in plants. And like we've done with all of these associations, some of them are good or mutualistic, and some of them are antagonistic or negative interactions. And when we think about the plants and their microbes, we should really be mostly thinking about the roots and the soil, okay? And so you remember that term we defined long ago when we had that soil lecture, and we defined a term, the rhizosphere, which is the region of soil directly impacted by plant roots. And this can actually be a very large area, particularly when you have like rhizomes spreading plants like grasses that often send out shoots under the ground. They can go on for miles and miles in association. So you can have microbes on the surface of the root, but that's actually fewer than 10% of the total microbes. So much of them will be in the surrounding soil, but not necessarily touching the root surface. And so this is kind of the rhizosphere effect, where you often have orders of magnitude higher numbers of bacterial cells, primarily bacteria, but other microbes as well, around the roots. And so what I want us to do is go into a breakout. And what you're going to do is basically discuss this. Talk with your group about why you think there's such a strong abundance of microbes that forms in association with roots. So let me make the breakouts. All right. Create. Okay, so take a few minutes to go ahead and discuss that, and we'll come back and talk about it. We're just waiting for a couple of students to come back, and then we'll get going. Awesome. All right, does somebody want to share what you talked about in your group?
My group talked about how roots are like able to absorb them, and they like take advantage of the nutrients and like water sources. And we also said that maybe they grow by the roots because like biofilms are able to be created where places are like moist and they can stick to the surfaces of the roots.
Okay, so that last part would be a part of that rhizoplane, right? That there might be biofilms that form on the surface. And so yeah, and you can kind of see if I reshare, right? So you can kind of see this is a root tip with some fluorescence microscopy, and you can see they live in association with that. So, and then you also mentioned kind of the absorption of roots of water and nutrients, and so that can benefit. In some cases, you could actually end up with a competition kind of between the microbes and the tree roots for certain nutrients, and we'll touch on that a little bit later in the lecture. Other groups have other ideas? What about those that aren't living right on the surface? What else might be going on here?
In our group, we spoke about how some microbes may be given like a microenvironment by the roots, like a small niche for a particular species. And another thing I thought about is like some fungi may break down some molecules for the plant for easy absorption, like provide all the nutrients.
Yeah, and that's not just fungi, right? So bacteria and fungi can both play an important decomposition role, and that can liberate things. Carbon decomposition isn't as big a deal because, of course, a plant is photosynthetic, and so they fix their own carbon. But decomposition of nitrogenous compounds and things like that can play an important role too. So yeah, I'll buy that. What else do you think might be going on in terms of a benefit towards the bacteria living near a root? I guess my question gets at, do you think this is a one-way street of stuff moving from the outside into the root?
It's a mutualistic relationship that can be established, possibly.
Okay. And so what kinds of things do you think the bacteria outside the root might be getting from the plant roots themselves? Let's see, somebody put in the chat. So Jenny mentioned nitrogen fixation, and we're definitely going to talk about that. So nitrogen fixing symbioses are really important. But as we'll see, those are often in like organisms living inside the root. For those outside, though, it's important to note that roots are leaky, right? So things get into them, but also things get out. And so remember, photosynthate, all of the sugars that are created up where the plant chlorophyll is, right? And all the photosynthesis occurs, those all get transported down to the roots because the roots have to be fed, right? So that they can divide and grow. But what can happen is the carbon and the nitrogen that's in the plant itself can leak out through the roots and create kind of this halo effect where you end up with the growth of bacteria being amplified because of that. So it really is kind of a two-way street. So that's good. Did anybody else talk about anything we haven't discussed so far? We'll definitely come back to the nitrogen fixation though. All right. So that's essentially what I'm saying here. I'm going to call it exudate, right? So, and in fact, it's not that the plants want to do this, right? It turns out that microbes can trick them into doing this. So microbes can release chemical compounds like hormones, which interact with the plants, cueing the plants to release these exudates and allowing them to feed the bacteria, right? So this is going to help the bacteria grow. It provides a range of things from vitamins to, building blocks like amino acids or sugars. And tannins and alkaloids are two compounds that are very commonly produced by plants. These can actually be deleterious or negative to the growth of other plants, and they may be bad for other microbes in some cases, but some microbes have adapted to feed off of the tannins and the alkaloids. And an interesting side note is that as a plant grows, its rhizosphere grows and matures. And so you can end up with a succession that occurs as the plant gets bigger. So when you go from a seedling, you're going to have a very different root-associated microbiome than like a mature oak tree or something like that would have. Okay. Now it's not all like give and take by the microbes. The plants usually benefit too. And this was alluded to by some of your comments. So for example, nutrients can be provided to the plants that otherwise wouldn't be available. So an example of that is the mineral apatite, which is high in phosphate. But plants can't typically break down the apatite mineral themselves. But there are microbes that through their activities can solubilize the phosphate, liberating it from the rocks in the mineral form and making it available. They can also produce different vitamins and amino acids that get shared over to the plant. And then these hormones are plant hormones, but they can actually be produced in part by the microbes that associate with them, or can be supplemented by that. And these plant hormones typically affect the growth of the roots and the leaves and the branches, etc. But in that local rhizosphere zone, they often will impact root growth. And so this can be a positive for both organisms. The other thing is that the microbes that are living on the surface of the root provide a barrier to penetration by potential pathogens. So if there's a biofilm on the outside of the root, it makes it harder for the other bad guys to get in. And then, as I mentioned, some of these compounds produced by the plant can be allelopathic or make it so that other plants don't grow nearby. Because of course, if you have two plants near each other, they're competing for resources, right? Even if they're a different species. But the microbes can also participate in that and produce some of these, chemical compounds, these allelopathic compounds that will limit growth of other plants so that it keeps their host plant happy. And then this is what I alluded to a few minutes ago, that it turns out though that nitrogen, it's a two-way, it's a love-hate relationship kind of. There are nitrogen providing associations that microbes give to the plants, but a lot of free-living microbes, they need nitrogen just like the plants do. And so if you have a root that's surrounded by nitrogen utilizing bacteria, or archaea for that matter, they are competing for that same nitrogen that the plant would like to take up and use for its metabolism. And of course, plants have a very high need for nitrogen and phosphorus. So if the microbes immobilize or utilize that nitrogen, then it can make it so the plant isn't getting that. So it's kind of a love-hate relationship with the rhizosphere. All right, so let's talk about nitrogen fixing associations, or the technical term. Remember, nitrogen, N2 gas, right? N2 is a azide, right? And so diazotrophy, right? Two nitrogens eating two nitrogens, right? That's how the etymology of that comes about. But basically, they're consuming N2 and turning it into usable nitrogen. So what do you guys know about nitrogen fixing associations before I tell you what I know? What are some of the things you've learned in other classes or in other contexts? What kind of plants tend to have these nitrogen fixing associations? Does anybody know?
Some crops, like beans.
Okay, so beans, or what we'd call legumes, right? So things like soybeans, things like, but there's a range of different things. Clover and some of these other plants have those associations too. All right, what else do you know about nitrogen fixing associations? Anything? All right, well, I guess we're going to learn something today then. Okay. So the first thing I want to start with is the reason that this exists. So we've talked a lot about nitrogen fixation this semester, and I think I may have alluded to this, but it's important in this context. Nitrogen fixation is essentially the conversion of the N2 gas into ammonia, right? And of course, as you know, ammonia can be used for a lot of purposes, but of course, it's a building block of amino acids. It's going to play a central part in a lot of cellular materials that require nitrogen, whether that's, and of course, we know there's nitrogen involved in much of the cell. But the key thing to focus on here is this high cost of ATP. It's really hard to split the N2. And if you remember, for the acetylene reduction assay, way back when, what did we talk about? Why did we use acetylene, and how was it similar to N2? Does anybody remember? Chemically speaking.
It had a triple bond.
It had a triple bond. So that's why it's so energetically expensive, because breaking triple bonds is hard to do. So you got to spend a lot of ATP just to convert one N2 into a couple of ammonia. And yet these microbes do it, and they really benefit their plant hosts by doing it. If you have a built-in source of nitrogen, then it turns out that it works out pretty well because, because what do you know about plants and ATP? Where are the plants going to get their source of ATP that they could share with their bacterial symbionts?
From photosynthesis.
Exactly. So photosynthesis produces a lot of ATP, as you guys learned about in general bio, right? As you go over in physiology course or cell biology class. So it really is a match made in heaven in terms of having a plant that has a ready source of ATP through, the Z scheme, and that ATP can then feed the nitrogen fixers because the plants have a high need for nitrogen. Now, the level of association is quite varied. So some of them are sort of free living, right? And there are diazotrophs that don't live on plants or in plants. Because of course, there are needs for nitrogen fixation of many bacteria as well. It's not just a plant phenomenon. But over time, what has happened is the plants have started to form these associations. And it may have started in the tropics. A lot of grass roots and some plants have associations with these. And if you see like azo or azoto as the first part of the name, that usually refers to a nitrogen fixers. One of the things you should know about nitrogen fixation is that oxygen is bad for nitrogen fixation. So the enzyme that catalyzes this reaction, who remembers what that's called? What enzyme does
Nitrogenase?
Nitrogenase enzyme. It turns out that oxygen irreversibly destroys nitrogenase. And so the organism would have to make a whole new set of enzymes. So it's often going to be in anaerobic environments, either where the whole organism is anaerobic, and in this case, like Clostridia, are fermenters that live under anaerobic conditions. Seagrasses, the marine sediments underneath seagrasses are anaerobic, so that's a good environment. But in many plant roots, right, where you have these legumes, for example, they're in aerated soil, so it's not necessarily anaerobic down there. And so that's how these very specialized nodulating associations likely evolved. And these are dominated by members of the genus Rhizobium or Bradyrhizobium. And so that's what I'm going to focus on this slide. So these are the legume-associated nodulating bacteria. And actually, the more we study these, the more we discover there's a diverse array. And so now there's like 6 or 7 different genera that are involved in this. Rhizobium was the first, but it's not the last, that's for sure. Now, these bacteria actually are capable of living under aerobic conditions when they're free living and not associated with their plants. And they have like a completely separate lifestyle when they're finding a new plant to associate with out in the soils. They are not fixing nitrogen at that point because there's lots of oxygen around. But when they find a plant, then they can form these mutualistic partnerships, and they can essentially invade the plant root to undergo this symbiotic association. It turns out that this has been very well studied because we like to throw lots of money at agriculture, right? Because it's involved in human food. And so we know quite a bit about a couple of groups, the Rhizobium and then later the Bradyrhizobium. So Brady just refers to like slow, right? Like bradycardia is a slow heartbeat. So the reason that they have a slow name is they're slow growers. And that's because it turns out that the genes that are involved in nodulation, the so-called nod genes, and the nitrogen fixation genes like nitrogenase, the NIF genes, in Bradyrhizobium, they're carried on the main chromosome of the bacterium. In Rhizobium, however, it's carried on a plasmid. And they can grow more quickly because they don't have to copy all of those nodulation and NIF genes every time they double their chromosome. So what this means is that you've got kind of a differentiation even among the different groups of bacterial symbionts here. But both groups end up forming these associations, and they form these round nodules all around the roots. And so when scientists first dug up, or farmers, right, they dug these up, they realized there were all these weird things. And if you cut open one of these nodules, it's filled with this red stuff. And it turns out it's the same red stuff that you're filled with as well. It just evolved completely independently. So we refer to these as leg or leguminous hemoglobin. And the use of the hemoglobin is totally different than why we use hemoglobin, right? Our hemoglobin binds oxygen to feed our cells. In this case, the leg hemoglobin is binding oxygen to protect the bacteria that are living there, protect their nitrogenase enzymes from the oxygen. And so what it does is it creates so-called microaerophilic conditions. It's not completely oxygen free, but it's very low oxygen. And that's a much more hospitable place to have a nitrogen fixation going on inside the nodule. So nodule formation has been studied pretty elaborately. And it turns out it's quite complicated, but it's really a delicate dance between the bacteria and the plant. And they have all these chemical interactions. And so it's a chemotactic based response where the bacteria have to find their plants, find the root, and make its way into the root, okay? And they have to recognize where to do that. And so this has evolved, co-evolved over millions of years, right? The plants do things that attract the rhizobia know what the plant is. So what happens is the plants will excrete chemicals like flavonoid compounds, and the bacteria find them, and they will release their own chemical compounds, and those are the nod factors or the nodulation inducing chemicals. Okay, so this will start the process of creating the association, whereby the bacteria can actually invade the root hair, right, via something called an infection thread, which is basically a cellulose tube filled with bacteria lined up end to end. And so what you can see here is if this is a root and this is like a root hair, then what happens is at the tip of the root hair, the bacteria find their way in between the cells, right? And they form the infection thread, which is essentially a bunch of bacterial cells making their way through this cellulose tube. And eventually then they find their spot inside the root, and that's where they'll form the nodules. Okay, so this just diagrams what I already told you in terms of the release of the plant factors, attraction of the rhizobia. It turns on, these cue the production of the nodulation factors, right? So it turns on nod gene expression, releasing those nodulation factors, which cues all of this morphological change in the plant. And ultimately, you end up with what's called a bacteroid, which we've encountered before when we talked about some of the animal symbioses. It's not a free-living bacterial cell anymore. It's now a modified cell which is inside the host plant. And it divides, but it no longer is free-living and dividing. It's propagating, and you end up with these sorts of modified rounded bacteroids. So it starts off as more of a rod, and then it turns into these round bacteroids. And what you can see is it's filled with nitrogen. These are nitrogen storage granules from all the nitrogen fixation that they go through. And then they're going to pass the nitrogen storage on to their host. And so what happens is the plants continue to divide their cells in the nodule, and the bacteroids divide and reproduce, and you end up with that leg hemoglobin filled, circular, spherical object, which is we can see with the naked eye as a nodule filled with thousands to millions of the nodules. Okay. All right. So let's have our first poll question to see how much we were paying attention. So which of the following would be true about nodulating symbioses? They're important for carbon fixation for the plant. They involve physical transformations of the plant, but not the bacterium. They involve chemical communications between the host and the plant, or I should say the plant and the bacterium. I don't know why I said host and plant. Both B and C, or E, all of the above. Let's go ahead. All right, is that everybody? Go ahead and log your answer. Okay. All right, so let's see. Most of you put D. Okay, so let's take a look at each of these. They're important for carbon fixation for the plants. What's wrong with that statement? What's going on here? Is it carbon fixation?
No, nitrogen.
Not directly, right? I mean, of course, it'll have an effect on carbon fixation more broadly, but really what we're focused on here is nitrogen fixation. So A was not a correct answer. So let's look at B. They involve physical transformations of the plant, but not the bacteria. So let's ask the first question. Does the plant go through physical transformations?
Yes.
Okay, and what are those?
It builds that infection thread, which leads up to the bacterium.
Okay, and then ultimately you end up with a big bulbous nodule, right? That's mostly plant material, by the way, right? Do the bacteria undergo a physical transformation though too?
Yeah,
What
they
did
turn
we-
into little round, like orbs instead of the
Right.
rods.
Right, so they start off as a rod and then they become a circular or a spherical coccoid shape, right? So B is incorrect because it's both the plant and the bacteria that change. So that means that D and E are not correct as well. So the best answer was C. There's chemical communication. The flavonoids and other compounds get released, and then the nodulation factors get released by the bacteria back at the plant. And so really C was the best answer. Does everybody see that now? Okay, and I need to change that. So it's between the bacterium and the plant. All right, any questions about those associations? All right, so now I'm going to move on to a different kind of plant microbe association, which is completely different, yet it still involves important nutritional elements for the plants. You have to keep these straight on the final. Rhizobial associations are bacteria-based symbioses with the plants. Mycorrhizae, as the name implies, are fungus-based. So myco, whenever you see that, you should think fungi. So this is a completely separate, yet important, very important association of plant roots with fungi. And it turns out that this is likely responsible for the fact that we have land plants on Earth today. These mycorrhizal associations probably date back to when aquatic plants first came up onto land, because they really facilitate the nutritional needs of plants as they move onto the earth, which is really important when you don't have a lot of rich soil already made. So if you think about those earliest plants, they were basically coming out onto rocks, not a very hospitable environment. Now, of course, we have very lush terrestrial environments, but they still play a huge role. Like with the rhizobium, those bacteria with plants, these are highly co-evolved associations. Unlike rhizobium, it's not really limited to certain groups like legumes. In fact, as I say in the first line there, probably upwards of 80% of plants across the whole tree of plant life, angiosperms, gymnosperms, you name it, there's going to be some sort of mycorrhizal association. Similar to the rhizobium, you do end up with a physical integration of the microbe with its plant hosts. But in this case, these are not infection threads and bacteroids because it's fungi. So it's the fungal hyphae that actually intercalate and get inside the plant roots. What this is doing is it's making it so that the nutritional, in particular the nutritional niche space, is broadened. They have access to more food, so to speak, than without their fungi. And so really, you have this interplay, like with the rhizobia, it's going to often involve things like nitrogen, okay? But these are not nitrogen fixers. These are fungi that can acquire nitrogen from the soil and deliver it to the plant host, but they are not nitrogen fixing microorganisms. Okay, so that's an important distinction between rhizobia and mycorrhizae. They do get their carbon from their plant host, so that's similar in the bacterial and the fungal associations. The photosynthetic plant can provide carbon. And in this case, they provide upwards of about 1/5 of the carbon for the mycorrhizal fungi. So what you're doing is you're attaching fungi to the outside and the inside of plant roots. And so this increases the surface area of your roots, essentially. It makes the roots be kind of more efficient. And it also allows access to different chemical forms. So remember we said that bacteria can help break down apatite and things like that for phosphorus. Well, the mycorrhizal fungi can access different forms of nitrogen and phosphate and deliver it to their plant symbionts. This is really important in, as I said, on like rocky islands and things like that, which kind of mimic the early Earth, like the early invasion of land on Earth. But also forests, rainforests are really important. It turns out that rainforests don't have very good nutrition in the soils. Okay, so a rainforest, even though it has this lush jungle, much of that jungle is only possible because of associations between the roots of those plants and their mycorrhizal fungi. And just to give you an example, this is a maple tree, right? So this is a maple tree growing normally under the same conditions, and this is a maple tree where the mycorrhizal fungi have been removed from the equation. And you can see how unhappy and, you know, low chlorophyll, low growth. So this one's not going to survive very long, right? Whereas this maple tree is totally happy. So it turns out that you can divide mycorrhizal fungi associations into two main groups. So there's the exterior and the interior ones. The exterior Ectomycorrhizae, right, are very common in groups of angiosperms and gymnosperms, especially in temperate forests. And so, as you may be aware, you know, there's different groups, different lineages within the fungi. The one we're probably most familiar with are the basidiomycetes. This is where our mushrooms come in, right? But ascomycetes, or so-called sac fungi, are really common. And there's lots of them. They're very diverse. But for example, truffles, which you may have heard about, right? In France, they get pigs to dig up truffles from in the forest, and they're very valuable and sought after in fine dining. These truffles are actually fungi that are ascomycetes, but they're root-associated fungi. Ectomycorrhizae form what's called a pseudoparenchymal sheath, which is just a fancy scientific term for an exterior sheath of fungi all around the outside of the root. In fact, it can make upwards of 40% of the dry weight of that root. So this is a cross section through an ectomycorrhizal infected root. So this is the stele portion and the cortex, the living portion of the plant root. And then on the outside, you'd normally, if there were no fungi, you just have like the dead parts of the root. But here what you can see is there's layer upon layer of fungal hyphae, and they form this tight sheath around it. And it's different than what we saw with the bacteria and the nodulation, but like that situation with the fungi, we also see a change in morphology of the plant. What tends to happen is the roots are simpler, they are shorter, and they are dichotomous branching, meaning they split into what? If something is dichotomously branching, what does it form? Phylogenetic trees are typically dichotomously branching as well. So what does that mean?
2 branches.
2 branches, right? So this is an example. So this is a bishop pine. If you've gone to Mammoth and driven through Bishop, right, you've driven near some bishop pines, right, because that's where they're named. Or the Doug firs, which are really common up in the Northwest where I went to graduate school. So these are important conifers that are huge trees, right? And they often live in pretty tough conditions, right? So soils that might not support the growth of something that could be 200 feet tall. But the reason they can do that is because they have these nutritional symbionts. And you can see the modifications that have occurred to these roots. And the yellow, almost like, I don't know, I think of Twinkies when I see this for some reason. I don't know why, like a big covering on the surface of it. But you can see that it's modified the root, and all of that yellow stuff is fungal hyphae. So the ecto, or outside mycorrhizae. But they do penetrate into the root themselves, but they don't go all the way into the living cortex. Instead, they're focused mainly out here. Okay, so this is an important distinction with the other class of mycorrhizal fungi. Ectos stay mostly on the epidermis or the exterior. And so you can see the up close microscopic image, it penetrates in, but it doesn't go all the way to the center. The other thing that you may or may not be aware of is plant roots often are very dynamic, and they grow, and then they, they tend to get bigger and bigger and longer, but they also, the feeder roots kind of come and go. And what happens with these fungi is that they make it so that they're more long-lived, the feeder roots. And essentially what happens is, if you notice, there's no root hairs coming off of this. Instead, what's coming off of these dichotomously branching roots are hyphae. And so you replace those root hairs or rootlets with hyphae, and they spread out all throughout the soil and bring the nutrients into the plant. So you're expanding the nutritional sphere, sometimes for very long distances in the soil. The other thing is you could see from that picture of the Twinkie-like roots is it's going to be really hard for a pathogen to get inside those roots and infect the roots. And it turns out that that's important because plant roots are the major source of infection. When plants get sick through disease, it sometimes occurs through a cut in the leaf or the stem. And we're going to see an example of that later today or early next lecture, because I don't know if we're going to finish. But oftentimes the soil and the roots are a major source. And so that's going to be protected as well. The other major group of mycorrhizae are endo or inside the cells. So they live inside the living cells. And so you can see the fungal mycelia fill, this is stained to light up the fungi, and they are much further in than we saw with the ectomycorrhizae. These can also be described as vesicular arbuscular. Many of them are in this group, which are the most common endomycorrhizae. They tend to look kind of like little trees, right? So there's this bulbous vesicle thing, and then there's like kind of a stem or a trunk. So that's where I think where it comes with the name vesicular arbuscular. Now, we said ectomycorrhizae were in the Basidiomycota or in the Ascomycota, but these guys are in another group of fungi called the Glomeromycota. And it's thought that these endomycorrhizae, which are much harder to identify because you don't see them on the root surfaces like you do with the others, these are the ones that probably date back tens to hundreds of millions of years. And we now appreciate, now that we've started like chopping up plant roots, looking for these things, and by we, I mean like botanists, right? And these kinds of microbiologists that are interested in this. We now know they're very common in angiosperms, gymnosperms, as well as early plants. And this is where I get at the point of them being important in the invasion of land. Things like ferns and bryophytes that have been on the planet hundreds of millions of years also have them. But they're much harder to spot because if you pull up a plant and just see the roots, you don't necessarily see all of the hyphae. Some of the hyphae will extend outside, but they don't form those parenchymal sheaths, so they're much more difficult to see. However, there is that connection between the inside and the outside when they're in the soil. And so they do provide very similar nutritional enhancements as the ectomycorrhizae did. And interestingly, those vesicular arbuscular guys, right, that I showed you a picture of, it turns out that there are bacterial symbionts that can live inside the fungal structures inside the root plant or the plant root. And so this is another example where you could have a tripartite or three-level association going on, where you have bacteria that benefit the fungi that benefit the plant. The other major group, which are less common, but are found in some plants like azaleas, which often bloom at this time of year, so we see them as we walk around. Or rhododendrons, or up in the mountains, you can see madrone trees. These are called ericoid endomycorrhizae. And so these are very specialized on this one group. And so it likely evolved once in this lineage and has just stuck with that. And these are characterized by having these very diagnostic coils that form inside the roots. So it looks a little different. All right, so understanding what the role mycorrhiza plays, you really have to kind of try to get at the big picture here. And so this is a paper from about 6 or 7 years ago now, where they were trying to get a handle on what the ectomycorrhizae, that would be over here, and the endomycorrhizae, those would be here, are doing, and how they're different. And so what this study showed was that you got to look at the flow of nitrogen and carbon. As we've talked about, carbon is going to come from the plants to the fungi. Whoops. Okay, so the blue arrows come out, but it turns out it's more complicated than that, right? So the fungi in both cases are going to bring in organic material, but that's much more a thing with the ectomycorrhizae that have those exterior structures, right? So there's not as much organic material that will come through the fungi from the soil in the case of these endomycorrhizae. But interestingly enough, it turns out that, and this likely means that much of it will be used by free living microbes living in those forests in the soil. So when you have ectomycorrhizae, it actually is bad for like the bacteria mainly, but other microbes living in the soils, because a lot of that organic material makes its way into the plant. Whereas with arbuscular mycorrhizae or the endomycorrhizae, that actually is not as big a deal. And so you end up with the microbes getting a bigger share of it. So it's kind of an interesting thing because it can facilitate interesting competitive interactions between these kinds of plants, as well as their associated fungi and microbes. And it has a big impact on how much carbon gets stored in the plant versus in the soil. And that's something we care a lot about, especially with global warming, global change, because we want to sequester or lock up as much carbon in soils and plant material as possible so it's not up in the atmosphere. Okay, so let's have a quick poll question to finish things off today. So which of the following would be true about mycorrhizal plant associations? So let's, they are found only in conifers or gymnosperms, right? That was the term we used. They're primarily nutritional symbioses. The fungi fix nitrogen for the host plant. They're most obvious when you look at the plant roots, or most of them are obvious when you look at the plant roots, and they're pathogenic. Okay, so which one is true? Go ahead. Okay, go ahead and log your answer if you haven't already. Okay. All right, so most put B and quite a few put C. So let's look at it. So nobody put A. So they're not only found in conifers, you got that. There's gymnosperms, angiosperms, all sorts of ferns. Okay. All right, B, they are primarily nutritional symbioses. This is a correct answer, right? So you're providing the nutrients from the soil into the plants, and then the plant is providing the carbon nutrition to the fungus. Why is C not the correct answer?